Okay. Thank you for that wonderful introduction, and I'm really happy to be here and really enjoy the conference so far. I won't be talking about matrix factorization, although it's near and dear to my heart, and I really enjoyed the talk this morning. But this talk is actually, it is similar in spirit in that a lot of what my group works on is trying to find kind of a concise representation of the data. No surprise to anybody here, we have a lot of epigenetic data. So if you consider a single epigenetic assay, we can represent it as a nucleotide resolution track. We have 3 billion base pairs of data, give or take. Okay, some of them are zeros. But we, of course, have lots of different assays, different contexts. You add the species, and really this blows up pretty quickly. So the question we're trying to answer is how do we actually capture the salient features of the data? And it's important to remember this is going to be context dependent. Maybe for whatever you're doing, you really need the nucleotide resolution, and maybe I don't. And we can kind of adjust depending on what kind of questions we're answering. So I'm going to talk about 2 different projects in the lab that address this question of how to compress epigenetic data in 2 different contexts. One is this was a GLI proceedings paper. It's titled Unify Hypothesis-Free Feature Extraction Framework for Diverse Epigenetic Data. And this is a method for describing epigenetic tracks in succinct representations. And in the second part, I'll talk about the differential loop excursion model, which is kind of a compression type model for high-C data. And as we compress the data and we make it smaller, we're going to get rid of a lot of features, but actually you'll see we might learn some new things. So just to start off, we can think about, well, epigenetic data is really large, but of course, if you think about RNA-seq data, you can also represent it as like a nucleotide resolution bigwig. And sometimes we do that and we look at it that way, but most of the time you're not actually analyzing the data in this space. Most of the time when you're working with RNA-seq data, you're actually looking at counts that have been aggregated over exons, isoforms, or genes. And this is, hugely facilitated by the fact that we all basically agree where the exons and the genes are in the genome. Now, if we take this analogy and I look at the epigenetic data, things get more complicated. We can agree that there's certainly features in the genome, and we have enhancers, promoters, gene bodies, transcription end sites, heterochromatin regions, insulators. Which of them is going to be relevant to any particular data is not immediately clear. These are highly context and assay dependent, whether or not you can even see them or consider them to be features in your particular data set. So how do we deal with kind of featurizing this data? So one really simple approach, we don't want nucleotide resolution data. It's very noisy, and that's not kind of the focus of our study. One thing is you can just bin your data. And so this is a pretty standard thing to do if you're working on some kind of downstream ML type application, but of course it's extremely naive. You're actually not respecting at all the inherent structure of the data. So we also have these kind of multivariate featurization techniques, chroma-HMM and various extensions thereof that are quite powerful at finding these multi-assay, highly reproducible genomic features. And of course, finally, we have, for single tracks, we have peak calling, which is quite effective in many cases, but can also be limited depending on the assay. So to give one example, some data actually doesn't really have these kind of narrow peaks that the methods are designed for. Some data comes in broad peaks. So these are broad regions of like constant signal that cover actually multiple genes. And you can actually still kind of try to find them by using MAX. You can run MAX in broad peak mode. And the way it does it is just a really kind of heuristic thing that is done on top of the narrow peak finder, where you find narrow peaks and you just try to merge them. So it's very naive. And of course, we can move on to something that actually is neither narrow peak nor broad peak. And actually, I'm not even sure what this is, right? So this is a ChIP-seq of RNA polymerase II. Of course, this is a really central molecule in gene expression. We want to understand what it's doing, where it's located, and why it has a certain concentration in that particular the genome. But it has a really complex pattern. You often see pretty sharp peak at the start of a gene. There's maybe a broader peak kind of near the start of the gene. then there's a very long, what I would call broad peak that covers the entire gene. Maybe then there's like another sharp peak at the end. So that's kind of like a prototypical polymerase track there. There's some variation, of course. So well, the question we set out to find, is there kind of an efficient algorithm that can just deal with this in a unified framework, just all of these kinds of tracks, and is going to produce a reasonable, meaningful interpretation, and capture the features and actually tell you kind of in a very agnostic setting what the features are. And so the solution we've come up with is to use L0 segmentation. So this, we believe it actually fits a lot of the requirements. So it is a fast way to segment linear tracks, and it can identify complex patterns. So the way linear L0 segmentation works is we're going to try to approximate your signal as piecewise constant. And so we have, just to take us through the objective, we have our data, and we're going to fit it as a piecewise constant approximation. And so we have some kind of reconstruction objective. So here, this is just derived from a Gaussian likelihood, but we also consider other probability distributions. And then we have a penalty term, which is, maybe you've seen fuse lasso, it's a very similar kind of penalty term. So we're penalized the difference between two adjacent approximations, but instead of penalizing it with an L1 penalty, which also would give you a piecewise constant solution, we actually penalize it with a L0 penalty. And this basically, effectively what it does is it just counts the number of jumps that your approximation makes, and it doesn't care at all about the magnitude of these jumps. This also makes the problem non-convex, but thankfully you can still solve it quickly, and it makes a lot of difference that this is L0. Just to kind of give a brief illustration, so this is just simulated data, so maybe you can actually already buy ICs. So there are 3 segments in this data, because that's how it was simulated. And so the approximation we're making is that like each of these segments actually represents repeated draws from the same distribution that is parameterized by some mean. So in this case, is a Gaussian distribution parameterized by theta. And so what I'm going to do is I'm going to replace the entire segment and all of these noisy observations with just theta. That's what the algorithm is going to do. And so, right, so exactly how much segmentation occurs is determined by this lambda parameter. And there's not really, you can use cross-validation to set lambda. I'll show some examples of what it looks like when you use cross-validation. But we don't actually think of this as something where there is a right answer. And instead, kind of when we look at different tracks, we usually look at a variety of lambdas to kind of understand the behavior of the data and help us also reason about it. And so actually, even the value of lambda itself is not particularly interpretable because it depends on the scale of the data. So what we instead like to use is this notion of compression ratio, right? So as you increase data, as you increase lambda, your data is going to be approximated with fewer and fewer segments, and eventually it's just going to be one segment, or just like this is the mean of the entire data set. And so regardless of what lambda is, I can always think about this kind of more meaningful quantity, which is like how much have I compressed the data? So how many segments do I have relative to the total length of the data set? So just kind of to illustrate a little bit about how this algorithm performs on some real data. Now this is just lambda selected with cross-validation. So here's CTCF. We have some genes on the bottom there, which is going to give you a sense of scale. The data is actually counts, but it's plotted on a log scale, which is why it looks a little funny. So yeah, it finds CTCF peaks. I mean, maybe, I don't know, maybe it doesn't find all of them, or maybe it finds too many, but it's, I feel like, approximately correct. So now we can look at a broad peak type structure. So this is H3K27ME3, and here it gives us these really nice step functions, right? And so this is something that indicates silencing a heterochromatin. So we can say like, oh, this gene is expressed, this gene is expressed, this gene not expressed, right? It has a high 27ME3. So, okay, well, same region. Now we go to Pol 2 in exactly the same location. And you can see here, it actually, we said it's a complex signal. We do actually get a complex pattern. So we have some sharp peaks, we have some broad peaks, we have some more complicated structures, and I'll come back to that in a second. So so we're definitely not the first people to try to do this. And one kind of key property of our method is that it is crazy fast. And that is not even really exactly our achievement, because actually all we did is we made a nice implementation of this paper, which is actually a really brilliant paper, but somehow maybe not so well appreciated. It also contains the fastest known algorithm for L1 segmentation. The nice thing about this algorithm is that while the problem is not convex and has square complexity in theory, in practice it is linear complexity on any kind of reasonable data, as is shown there. And we're beating all of our competitors by wide margins in terms of running time. So one of the things that you can ask is, well, how well are you recapitulating kind of this part of the data that we care about? So we have lots and lots of evaluation in our paper. I just give you kind of like a flavor of the kind of evaluations that we do. So hopefully you're familiar with this kind of chromo HMM type annotations. So these are these ways to paint the genome with classes. And they have names, right? We have like TSS, right? Where did those come from, right? Chromo HMM is an unsupervised method. It does not name anything. You have to name stuff afterwards. So, but this process has actually been kind of automated, because the way we name chromo HMM classes is you just kind of go back to either the data was used for it or in some other data. You just say like which track is enriched in this class, and then based on those patterns, like promoters are enriched for K27 acetylation and open chromatin and maybe polymerase. So you get a kind of like enrichment pattern that's associated with a particular class. So that's what allows us to have these like very stereotyped class names. If you look, all of the roadmap data sets use exactly the same class names. And so one of the things we can check is like, what if you did this enrichment not on the data, but on the data that we segmented, and we segmented it actually 10,000-fold. So this is a huge data reduction. And basically, this is what's showing is that like you actually get a very, very similar result. So you've lost very little by compressing all of your data, which actually comes in diverse types, broad peaks and narrow peaks. We apply exactly the same compression ratio to all of it, and all their salient features are retained. In contrast, if you bin the data at 10,000 K, you would lose all your signal. So, okay, I just wanted to get back to this, this kind of interesting example of the polymerase, which really has a complicated structure. And so if you go to like a standard epigenetic repository, what you're going to get is a kind of a post-processing. You can get narrow peak calls on this kind of data. And the narrow peak calls they will find the TSS, right? So we do actually have a nice narrow peak at the TSS. This is something that Max is very good at finding. Often you have more than one peak. It may be actually quite complicated. So it is definitely, if you're interested in the TSS, you can use Max and you can get a lot of information. But there's actually a lot more information in this data. So for example, one thing that you can note is that there's a very, subtle, maybe difficult to appreciate by eye drop off at the transcript end. Right, and this is, polymerase is coming off here, so that makes sense. It's done transcribing the gene, and our framework actually picks this up, but it's not a peak; it's a breakpoint, right? So it's if you're sometimes there's actually a peak at the end, but more often it's just a drop off. It's like cliff. So a peak finder is actually not at all set up to even answer this question. And we can actually turn this into an evaluation where, like, we can ask, for our breakpoints, how good are they at calling transcription end sites? So for this evaluation, you want to be here. We can use Max as kind of like a trivial tool that we know is not doing particularly well on this task. And so the reason I have this figure up here is to point out that we have all these different kind of versions of segmentation. We have the L0 versus L1. We have the Poisson versus Gaussian, and to do well on this task, you really need the L0 and you need the Poisson. So you really need the complete full formulation to do the best possible on this task. Okay, so just to kind of point out one last little fun aspect of this approach is, okay, so we have some kind of start. We have the gene start here. We have a sharp peak. We have the transcription end site here where we have a level change. Now, that's actually not the only level of change here. There's other things going on. So, for example, we have this thing over here. I mean, maybe that's an artifact. I don't know. I mean, certainly this method is perfectly capable of like placing a breakpoint where nothing's going on, right? It's just a statistical approximation. But actually, we quite reproducibly see this, and it's, of course, very nicely corresponds to approximate the location of the first splice site. And this is not actually, if you look in the literature, this corresponds to something that's really well known in biology, is that the splice site increases the efficiency of elongation. So what's going on here is that, the polymerase, what can it do? It can fall off or it can go forward. And basically what's happening here, you can't tell us from the data, this is kind of the synthesis of the data and the kind of our understanding of what's going on, is it's going faster, right? So we're detecting it less because it's going faster. And I think this is actually kind of maybe an important thing to think about. I mean, the polymerase is a really large complex. It's really important. It interacts with like anything else around is going to have to somehow interact with it or move past it. And thinking about like where it is at any given moment is obviously really valuable. And we kind of get this information by doing ChIP-seq. But there's also a whole extra dimension to it, which is like it's not just where it is, but how fast is it moving, right? It's a motor protein. So when you see less of it, it can be because it's fallen off, or maybe it's moving faster. And maybe how fast it's moving is actually a more important quantity, right? So we can think about the flux of the polymerase through a particular location. So, and this kind of representation actually, I think, really facilitates being able to think about this. I mean, I just encourage you to like do this like mental experiment where you imagine this red line isn't here and just try to find that breakpoint from the black dots. I think it's really quite hard. So I think this is currently we're actually thinking quite a bit about this. So I've managed to explain this thing. What about this? I'm not totally sure what that is. I would like to hear some ideas. As just to show like another gene, which this one has a really complicated TSS pattern, which has to do with like where polymerase binds, but have a very nice splice site here for the breakpoint. Okay, and just kind of a final note about this approach is we also can handle DNA methylation, which is a little bit of a special track in the sense that the way the data comes is a double track, right? So you don't just have read counts for every location. You actually have methylated and unmethylated reads. So you have two values for every location. And this is actually quite naturally modeled with the binomial distribution. So we can run exactly the same algorithm, just assuming a binomial distribution and formulating this loss. It is actually this turned out to be like numerically kind of a difficult problem. So the algorithm is slower, but turns out it's fine because there's not that many CPGs, right? If you just care about methylation and CPGs, it's way less than like every nucleotide, about I think 70 million CPGs. So overall, I think the total genome segmentation running time is about the same. And just to illustrate kind of how methylation segmentation works, so we have our this is a locus. This is an OCT4 locus and H1 IMR90. So this is a gene that's expressed in H1. It's a pluripotency gene, so it's expressed in stem cells. And so you can see the yellow bars up there. Those are that's the methylation track, but the two tracks have been collapsed down to ratios, right? So these are now the beta. So we don't actually know. Like we can think about it as like either two sets of counts or like a ratio and a coverage. So I'm just not showing the coverage here. And then we have like a couple of different segmented versions. So you can see kind of how as we like segment this structure more and more into fewer, as in like we compress it more and more and we segment it into fewer and fewer segments, you can see how it evolves in terms of what it thinks the salient features of the data are. And naturally, so one thing is L0 is much better at doing this than L1, and so naturally, like one of the features that you see is that in the H1, the promoter, which is hypomethylated region, this being an expressed gene, is very naturally identified as a kind of unique methylation feature that's distinct from its neighbors. all the way up to fairly high compression ratios, up to, so for methylation, we have 1,000 here. But another kind of interesting aspect of this is that in this setting, where this gene is not expressed, we still actually managed to identify the promoter. So the promoter is still special. I mean, the level change is much, much smaller, but from the methylation perspective, even in this like non-expressive setting, the IR90 cell line knows where the promoter for this gene is. Right? It has a special epigenetic property. So just to kind of wrap this up, so we have a nice fast implementation. This is a link to the software package. So we've designed it kind of thinking about epigenetic data. So it will work directly with bigwig files. We have Gaussian Poisson, and we have implemented binomial for segmentation. We are also throwing in our super fast implementation of L1, so also known as fused lasso, just throwing it in there because it was in the same paper, though I think the algorithm is actually completely, well, yes, very different, I'll say. And our implementation, we do support observation weights. The thing I like to point out is like, yes, we thought about genomics. It's not actually genomic specific. Like if you just have like a vector of data, you want to segment it, you can just throw it in there. And one thing I'll promise you, I don't know about the results or they'll be interpretable, but it'll be really, really fast. Okay, so that was all kind of trying to understand the 1D genome, but the genome is not 1D, right? Actually, it's 3D, and we already saw one of these figures earlier today. And the 3D organization of the genome is really important because that is the textbook picture of how genes are regulated. So genes are regulated by regions that may be very, very far from the promoter in terms of where they are in the chromosome, but they're nevertheless able to influence the gene expression because in 3D space, they're right nearby, and in this case, really actually interacting and forming a complex. So real genome is 3D. But most of the time, actually, we think of it as being 2D, right? Because the way one of the most kind of effective ways that we have of measuring the 3D interactions is by doing these collocation assays. It's not the only way, but that's kind of the one where we have the most data. And what that produces is this kind of contact map. Hopefully, this is familiar to everybody here, but just in case it's not. So this is a contact map is ordered just linearly by chromosome position. And each like IJ cell represents the frequency of contacts between those two positions. So naturally, anything that's on the diagonal is actually literally physically linked. So that is ligated very frequently. But then once in a while, we see these really high ligation frequencies for regions that are not in physical linkage on the chromosome. And that represents exactly this situation where they're actually contacting each other in 3D space. So we've had these contact maps for a while, and we've actually developed a robust theory of where some of these features that we observe in the contact map come from. So one of the most prominent theories, I'm calling it a theory because I've been told not actually everybody agrees with this, but it is winning. It is a winning theory about what is going on in the genome, is the loop extrusion theory. So the idea is that we have this motor protein called cohesin, and it has two legs, so it can bind to DNA, and it will extrude a longer and longer loop. So it will kind of walk along a DNA in opposing directions and create this loop, thereby bringing distal regions together. And so this is constantly happening, and it is a very dynamic process. And actually what we observe, when we observe this is actually an average over time. So of course it's an average over cells, and those might be like very heterogeneous, but even if you had most homogeneous cells, you would still observe this according to loop extrusion, because this is you haven't frozen the cells in time. So maybe the process is actually the same, but you've captured the loop extrusion process at a different point in its kind of lifespan. So, okay, so there's a lot of people that have been really thinking very hard about the dynamics of loop extrusion and kind of how to simulate it and really understand this from a physical perspective. And so for our model, we're actually very much inspired by the work of Jess Frudenberg, who kind of created this concept of doing, you can try to simulate the chromatin in 3D, but then we have this 2D data. Well, what if we just do the simulation in 2D? This actually simplifies things quite a lot, right? So, so the way you do the 2D simulation is you have a cohesin, and it starts out on the diagonal, right? It starts out where both legs are in the same position. And at any point in time, it can do nothing. It can move one leg forward to make the loop longer by one in either the kind of the right or the left direction, or it can detach. And so you can simulate this process with Gillespie simulation. So I simulate a single cohesin, and it kind of walks along. I put in some CTC outsides here, so it gets stuck at a CTC outside on one side and spends a lot of time there, and eventually it falls off. So each kind of the color here indicates how much that one cohesin would contribute to your final contact map based on its residence time in that position. And so we can add a second cohesin now. So that one did something a little bit different because like I'm choosing these things randomly. And then I do this a lot, and I get a TAD, which I've created by placing these control points, the CTCF points. And basically what those are doing is that they're really lowering the probability that the cohesin can move past it. The probability is not 0, so I still fill this out, but it is small. Okay, so what we propose is the differential loop extrusion model. And basically, this is a model that captures what this simulation looks like at the steady state. After I just run it for a long time, what does this actually give us? And so we have this notion of the physical process, and that we have this cohesin loop, and all it can do is move forward or fall off. And the rates at which these things are happening may be position dependent. And so we can think about kind of a particular cell in the high C. And so where does this come from? Well, this is a loop that's just longer by 1 than a previous loop. So it has to come from here or here. And then in the next step, where can it go? Well, it can go either here or here, or it can just kind of detach and make no further contributions in the contact map. And so this actually gives you a very simple expression for the steady state concentration of this cell in terms of its neighboring cells and a few different rate parameters. So I'll go in a little bit more detail. I think the model will become a little more clear. I just wanted to mention kind of one thing. I'll be showing a lot of high C pictures, so I think it's important to kind of know what we're looking at. So often when we're looking at high C, and that's kind of how I showed it in the very first slide I mentioned it looks like this. So it's not negative because it's like a contact maps, contact frequency. But sometimes we also kind of see pictures like this where all of a sudden, red is positive and blue is negative. Like where do these negative numbers come from? What does that mean to have negative contacts? So these are so-called diagonal normalized or observed versus expected. And so all we've really done is, so we've taken this is already in log space, just otherwise it's very hard to see because the diagonal is so dominant in this plot. So we just subtract out the diagonal values. And the reason for doing this is partly visualization. Like you can see, this is exactly the same data. So you can see a lot more structure in this representation because so much of the variance of this data is actually determined by this diagonal decay. So if you remove it, you can see a lot more structure, a lot more looping structure. And so we kind of have this, we thought about when we fit models, well, which data should we really fit it to? On the one hand, the raw data is actual physical quantities, is some kind of concentration or at least proportional to it. But on the other hand, when you fit models to it, your model spends a lot of time trying to fit the diagonal decay, and it will be a while until it even gets to the point, if it can even do so, that it can fit the local structure. Whereas this is actually, it's not physiological, but you can see kind of a lot of detail, and it's exactly the kind of detail that we're after, right? Probably maybe this is a promoter enhancer interaction. This is what we want to know. So so I guess one thing we realized is that based on kind of this loop extrusion dynamics, we probably can't actually fit the diagonal at all. If you can kind of decompose the diagonal decay into a term that is a function of loop extrusion, and actually you can pretty quickly, just by the formula, see there has to have an exponential decay. But of course, like chromatin comes in contact and through processes other than the cohesion bringing it together. Like it can come in contact randomly based on like the polymer, like its polymer dynamics. And that gives you a power law. That's been something that's been worked out by multiple people. And basically, the power law is really dominant in this data. So there's really like a very small window where the loop extrusion actually even has a contribution to the diagonal decay, and it's still actually tiny. But anyway, so the solution is really simple. We have a biophysical model. It's going to run on the absolute scale, but before we like do anything, we fit it. It actually is going to self-normalize itself, and so we do all of our fitting in this space. And so we don't have to worry about the polymer terms. Okay, so I'm going to tell you a little bit more about how we fit the DLM model. Just go kind of step by step. So we can look at like a single entry in our matrix here. So this is our formula for how we get the value of that entry in terms of our parameters. So this is the entry we're looking at. It is going to be a function of its neighbors. So these are the loops that are shorter by 1. Times the rate parameters, so like how much the cohesion is allowed to actually move to extend the loop. And in the denominator, we have like the outflow parameters, which is how much the cohesion can move from this one to like an n+1 loop, either in the right or the left direction. So that's it. It's actually a very simple formulation. Okay, and so how do we fit the model? So we instantiate locus-specific rate parameters, left and right. So these are the green and the purple arrows here. So these are vectors. So these are we're gonna fit these. So just plugging them back into the equation here, you can see the colors match. So what our model is going to do is it's going to generate predictions. Given one diagonal, your task is predict the next diagonal in terms of these parameters. And so, right, and then we just compute MSE with respect to what we actually observe in the data. Now, one thing you might already kind of guess is that your MSE will get crazy small when you do this, because predicting the next diagonal ensures the previous diagonal is not a difficult problem. You can literally just average your neighbors and you will do an okay job, right? That's already going to be a great prediction. So it seems like maybe we haven't accomplished anything, but we have. So the way we compute the fit actually is we start from a constant concentration, and then we roll it out using these fitted parameters, right? So we got some kind of fit. These parameters are variable, and we're going to compute an output starting from constant. So here's my video for computing the output. It's amazing. Looks pretty similar, right? And this is starting from a constant concentration. So all I've learned from the data are the purple and the green rate parameters. That is it. So kind of another way to look at this is that this is my data. This is my 2D data. I've reduced it to a 1D representation. And to get the data back, I've actually completely hard-coded this particular decoder form, right? So it's not a factorization. This is not a linear function, but it's a very simple function. And so you just run it forward and you get your data back. So this is kind of an example of a really small patch. I think sometimes when people look at it, it's just like, why is this matrix not symmetric? Well, it's not supposed to be symmetric. This is the data, and this is the predictions. It's just in this particular case, they look so similar that you might think that I'm trying to show you a symmetric matrix and I messed it up. No. So, okay, so this is like a very nice small patch. If you go out to a bigger patch, it looks more kind of like this. And here, hopefully, you can tell the difference between what is data and what is the fit. One thing about the data is it is very kind of speckly and noisy, and the fit ends up being very smooth. So we can visualize the rate parameters, which we've learned from the data. And so you can kind of appreciate, so in these places, right, like we have these kind of TADs, and maybe you can call this like a TAD boundary. So we have like an insulator type element. And so the way our model kind of thinks about these insulator elements, and these are places where the cohesion has a very low probability of getting past. And we indeed, these are not rates, they're actually expressed as processivity. So like when you have low processivity, that's when you're getting stuck. And you can see like, right, yes, you're getting stuck here and here. And for this boundary here, we also are getting stuck in one of the parameters. So kind of it makes sense kind of with our intuition of what's going on. So just to kind of show a few more fits in terms of, these are pretty, I think, diverse, showing a lot of different features. And showing that this model actually, it's an extremely simple model, and it's really surprising, I think, to me to think about how it fits all of these different patterns. This is not a perfect fit, and there's certainly, you can point out to features that are missing, and I see them also working on that. But I think it's a remarkably good fit. So, here we can look at kind of, this is now a genome-wide fit. So this is just like how well we're able to reconstruct the data from our 1D representation. And so we compute correlations in the observed versus expected space, so not in raw space, like we're looking at the kind of the more difficult problem. And we're getting, I think, quite reasonable correlations in 2 different cell lines. It's about, these are all 2 megabase patches. And then, and one thing that we've tried to kind of, now that we have these parameters and we've reduced this 2D thing into these 1D parameters, well, now we can ask all sorts of questions, like what do these 1D parameters, are they related to any of the other 1D parameters that we have? Those we have lots and lots, like for H1, there's just so many tracks you can get. What's the relationship between these things? So one kind of simple thing that we did is we fit a multivariate linear model for predicting these processivity parameters in terms of encode tracks and a CTCF motif. And so this is kind of, we make a pretty sparse model, so it's not the best fit, but it's just kind of a nice sparse model that doesn't use too much data. And this is what we get. So we have a left coefficient. Oh, this is cut off, but this is a right coefficient. So one thing you can see is, like, for the most part, this actually get very similar. So one of the main predictive things is CTCF chip seek. This predicts your processivity. Coefficients are negative because when you have CTCF, your processivity is low. One kind of another sanity check that we can point out here, we have CTCF, this is not chip seek, this is CTCF M is CTCF motif. And the motifs are stranded. So we have like a plus and a minus motif. And so of all the coefficients, these are the ones that are completely distinct, right? Because the motif does affect processivity in a directional manner. So depending on which way your CTCF is pointing, it's going to affect your processivity from the left or from the right more. And more people have actually measured it. It's like a factor of 4 or 5. So so this actually also checks out. And we can kind of, so these are things that are expected, and what else is going on in this plot? So I think after we, checked off all the expected thing, then we have some kind of mysteries here. So one of the strongest associated thing is this track called CHG7, which is some kind of helicase. It is not really a encode track I've ever thought about before until I saw this plot. So it's not something that's been strongly implicated in loop extrusion or chromatin folding. If you have any ideas about why this is there and why this is our third hit for this process, that would be, I would love to hear them. Like I said, most of our parameters are negative. They slow things down. That makes sense. That's kind of the picture we have in our head. We have some parameters. They have positive coefficients. Again, I'm not sure where that happens. Oh, one last thing I want to point out. I was expecting to see this, and I was really happy that I did. K36ME3 is just a multivariate model, so it's independently associated with slowing down cohesin. So this is a great marker for active transcription. And I was expecting this because people have shown that, remember, polymerase, very important. It has been shown that actually can antagonize loop extrusion, right? These are motor proteins, they interact with each other. When they encounter each other, something's going to give, right? So polymerase seems like it might win and actually slow down the loop extrusion process or make the cohesin fall off. So, okay, so we have this nice compression model, but we were thinking, well, can we actually, this is not a predictive model. I can never tell you anything. If I fit something to one locus, it doesn't tell me anything at all about a new locus, right? All I can do is you take me a locus and I can compress it and give you this nice representation, but that's it. So it's not a predictive model. But of course, like, we have these really nice predictive models, and we were wondering, well, can we actually turn this into a predictive model? And we're very inspired by this C origami paper, which is not, so we have some purely sequence to function models where your predictive model just takes in sequence, and like Akita and Orca, those work very well. So this model takes a slightly different approach. They're still gonna, they're using sequence, but it's more of a contextualized model where you have sequence, but you also have epigenetic tracks. And sort of the rationale for using them is that we really want to focus on cell type specific effects, right? Sequence is the same in every cell type. So how we're ever going to produce like a cell type specific contact map, if you didn't give me any information about which cell type you're looking at, I need a piece of contextualizing information. So in the C origami paper, they use CTCF and an open chromatin track from the cell type that you want to make a predictive model for. So we're basically taking exactly the same approach. And so we have our we have our open chromatin, and we use sequence, but we actually use sequence in an extremely naive way, where literally we just orient the CTCF motif. We know orientation is critically important. It's not something you get from ChIP-seq data. ChIP-seq data doesn't know which way it's facing. So we basically use the sequence information to orient the ChIP-seq track. And you put it through, simple neural network, and output the left and right tracks. And once we have the left and right tracks, we can make the contact map. So here are some examples of how this works. Now, this is actually a predictive model. So these are some loci that the model has never seen before. It's making predictions on those loci just based on that limited information. And so just kind of for some comparison, this is like not a real evaluation because I've just taken a figure from the C origami paper. I mean, I think we're doing the same thing in terms of what's written, and I've just put up like our plot right next to it and lined up the axes. But we're actually doing pretty well, right? So this is C origami. It's using exactly the same data. This is our performance. It's not quite as high, but it's pretty good on completely held out data. And one thing about our model is that it only has 536 parameters. So you might wonder actually, like, why is that? That's too many. Like, I'm using like 2 tracks and some motifs. Why we have 536 parameters? Won't go into it now, but I'm happy to explain that later, why you actually need even that many. But it is way, way less than like a full deep learning model, which have like hundreds of thousands of parameters. And one kind of fun effect of this is that something that's becoming kind of like people are discussing more and more is just like, to what extent we have all these sequence to function models, but to what extent are they memorizing sequence? I mean, there's a lot of homology in the human genome, kind of short range, long range homology. So even if you do chromosome holdout, is that really a holdout? Because some of those sequences, you've literally seen them before. So you could have memorized them. And people make claims, I think pretty substantial claims that yes, it does. We do overestimate performance because of homology, which I actually think that's not even the main problem. I think the main problem when you have some sort of homology and contamination, you're kind of violating this mechanistic assumption. Like you're building sequence to function models saying like this sequence feature like really causes this function. But if you memorized it, that's not true. It doesn't have to be causal. Now it's just an association. So one thing, because we've used very low sequence, you can't accuse us of memorizing sequence. We've used almost no sequence. We just do CTCF motifs. So I think this actually a pretty effective model. So just kind of to summarize, so I've discussed some ways we think about compressing data and creating these reduced representations. And one thing is to do so, either maybe you need to understand something about the data, or maybe you will understand something about the data once you do it. And so like, obviously to create the differential loop extrusion, we had to have this idea of loop extrusion, which we didn't invent, right? Like this was this phenomenal idea. And this allows you to represent a complex data set in a much reduced form. But also, once we actually go through these procedures of reducing the data, we end up learning more things about the data, right? So for example, we can see that the splice site actually is increasing the efficiency of elongation. Something is going on with CHD7, not sure what, please tell me, but might be important. And yeah, I think this is a good place to end. I'd just like to thank. So this was a major kind of piece of somebody's Tural's here PhD thesis, and he is solely responsible for the L0 segmentation, and it was quite heavy lifting in terms of the math and the implementation, and he did an incredible job. And the DLM model is actually a collaboration between Tural again and a postdoc, Tina here, who is she actually comes from more of like the dynamic stochastic modeling world. So it's been actually a really kind of productive corroboration of these nicely orthogonal takes on data. And of course, I like to thank my collaborators and the funding, and happy to take questions.
We do have time for questions, so please.
Right, perfect. I'll be the one. Thanks for the great talk. One question, one quick suggestion. On the first part, when you're doing the binomial L0 segmentation on the methylation, I presume you're just assuming they're equally spaced, which is not the case, and they're scattered across the genome non-uniformly. Can you think of a way to account for CPG distances?
Yeah, so we can put in weights. So you can have observations which are weights on the observations, but if you actually want to kind of have a weighted segmentation, you can also instantiate individual lambdas it's not a constant lambda, and that we can also do. I'm just not really sure how to set it up exactly, right? Like exactly what should be the function that transfers like distances to a lambda scaling. But I think that is how you would do it. And it is like it's implemented. So if people want to play around with it, it's possible to do. So yeah, I mean,
generally most methods do this, but I wonder.
Yeah, I think like you make a really good point. And that's something we thought about. We know it's possible, but we don't have a great way to evaluate it. Actually, yeah, just evaluating methylation segmentations is already a challenging task. Like, how do I know that one's better than another?
Great. For the second part on the CHD7, it's a known chromatin remodeler. There's diseases apparently associated that I was looking at, but one thing maybe you may look at is whether it's associated with cohesin loading. Whether it's associated with loading of cohesin, right? There are transcription factors, chromatin remodelers, although they are not necessarily the loaders of cohesin themselves. They highly correlate with the higher loading of cohesin. So it might be one of those that's like.
Right. Yeah. So I guess we're not modeling loading at all. We're just assuming.
No, you can just look at cohesin ChIP-seq data or like cohesin loaders like NIPTO as well.
Right. Okay. So I guess. Okay, I have to say something. If you cohesin is not in this model, because literally, if you include it, it'll be like you're just predicting cohesin. It's a really boring answer. And so cohesin data, it doesn't just reflect loading; it reflects kind of the steady state concentration, which is a function of all these things. And we're modeling constant loading, and we know it's not constant, but we don't really. I feel like the model already has enough degrees of freedom. If you start introducing other things just get worse. In fact,
it's the rate of it is one thing, but also the locations at which cohesin is preferentially loaded is another thing, and then maybe CHD7 correlates with those locations.
Yeah, no, I mean, I agree. It's important, and there's a lot of parameters we would like to fit, and just either we don't have the data, or there's some kind of numerical instability, and like once we start adding parameters, things it's ongoing effort.
Really great talk. Thank you. I just had a question regarding the use of your model for ChIP-seq data. Do you mainly work with the track that exhibits full change over control? You don't work with signal p-value? Or have you tried to integrate both, or do you pick one or the other depending on the task?
I mean, I think we often use single p-value and full change over control, and I mean, they all work. I think it's hard to say exactly it's going to depend what are you looking for? Are you getting what you want? And I think that the biggest power of this model is when your data your data is complex, and you want to kind of just like a simpler representation, just so you can formulate some hypotheses about it. So obviously that's ultimately going to depend, on the user, like I do like what you're saying. But yeah, those we do use them. We've tried them. They do work. Yeah.
Do you, I think so. The second part of my question is there. Has your lab tested out different ways to integrate both in like the same model? If both types of representation of the data,
I guess something I've alluded to is having looked at this polymerase data and also knowing that we know it's actually polymerase, movement is important for 3D chromatin organization, and then seeing it again come up in DLM, in this different form. I think that's something we're thinking a lot about adding, it's a motor protein. Cohesin is a motor protein, so you can model these dynamical parameters. Polymerase is actually kind of similar, so you can think of it as just having these dynamic parameters. It wants to move, but then maybe its processivity is altered by in different states, and maybe that's kind of something that we've been thinking about adding that in. Yeah.
Oh, and just one last comment. I know. I'm sorry. I didn't see behind me. My apologies.
Thank you.
So I will have a quick one. So for your first method on the segmentation, is the background important for your method? I mean, it seems like you tested on the ChIP-seq data where the backgrounds are pretty high. But going forward with this new epigenomic technology like cut around, cut and tag, there's almost zero background. Is your algorithm?
Well, for Poisson, 0 is okay.
Sorry?
For Poisson, 0 is okay.
Yeah, I'm curious. If the starting
yeah,
point
and sorry, here, let me go back a little bit. So it's purposely exaggerated because this is on a log scale. So I think if you plot this on a linear scale, it looks like much noisier than it is because it's plotted on this log scale. If you look at it on a linear scale, it would the data is better now, I agree, but this is maybe a bit misleading. But yes, it's fine. 0 is fine. We love zeros.
Okay, yeah. Good to know. Thank you.
Very interesting parts of your talk, but so for the second part of the talk where you talked about the DLEM model, something that was not clear to me is where is the CTCF coming in? Is it coming? So DLEM is a model that is fitting the high C data, and then you're correlating how are you bringing the CTCF in by correlation?
Right, so we have these parameters, the left and the right, they're green and purple here, and so we predict those parameters. So we have this 1D data, and we predict 1D parameters. And this is really nice because we don't need any kind of attention. Everything is super local. And then from here, you just have this completely closed form decoder that turns it into 2D.
So you're just doing a regression or something.
Well, like I said, in this case, this is not a regression. This is actually, it is a neural network in this case. If you want a good fit, you basically have to.
So you're predicting the parameters from these, and then you're reconstructing.
I'm taking in kind of these local parameters, predicting a local processivity parameter for the cohesin, which has a left and a right, and then just rolling out the steady state simulation with these rate parameters, and that generates a contact map. And this is the data. This is the fit. Is that, I don't know.
Yeah, that's good. Yeah, but C again is okay. Does it generalize across cell types?
So, so this is a contextualized model, right? So this took cell type information. I mean, one thing we've looked at is predicting some differences across cell types, and then just do well at that. And that was the purpose of C origami as well. And that's kind of one of the things that we're after. High C is actually quite similar across cell types. The broad features are extremely similar. These differences across cell types relative to the overall scale are quite small, not to say they're not important.
Hi, thank you for the talk. I just have a very quick curiosity. I was wondering if you were to segment many genomic tracks in many individuals, how the boundaries, with the same parameters, of course, to what extent the boundaries of the steps would be consistent
Yeah.
across individuals?
Right. So, I mean, if you have really clean data, they're kind of consistent, but not exactly the same, which is obviously kind of problematic if you're trying to then have some kind of joint representation.
Yeah, that's what I was
And
thinking about.
So the multivariate segmentation problem, I asked many theoreticians about this, does not adopt a really quick solution. And
Is
so
it-
everybody just says, well, just cluster breakpoints, just like cluster breakpoints.
Yeah.
And because like if I give you a set of breakpoints, the optimal solution is really easy, right? Condition on the breakpoints is like, so.
Is it plausible to assume that there are some kind of genomics regions in which these boundaries are more consistent, in your opinion?
So I mean, I'm saying they're highly consistent, but I can't promise you it's going to be exactly the same integer nucleotide.
No.
Right. So you have to, I don't think I have a really elegant solution that is fast. The fastest solution is just post-processing, just cluster breakpoints and reassign them. Yeah.
Well,
And
great.
actually we have, for methylation, we've done exactly that because we do like to kind of work on multi-sample data sets in the methylation space.
Thank you. Great work.